Hi guys, I'm Kristen Urquidez. I'm a science teacher here in Bakersfield. And this week I wanted to do a demonstration with you about the cells in our body and do an investigation to figure out how things can travel in and out of the cell. And we've got a little bit of an Easter twist for it too, since Easter is coming up. So the way this investigation works is you have to pick something that is going to be very similar, as similar as possible to the cells in our body. You're actually made up of trillions of cells, and we know that when you eat food and you drink liquids, somehow those things, the food and the liquids, can provide energy for you and nutrients for your cells. So there has to be a way for that energy from your food and the nutrients that you consume to get inside your cell. So our model that we are going to use are actually chicken eggs. And I have been soaking them in vinegar for about 2 days. And the reason I did that was to remove the hard outer shell that surrounds chicken eggs, because our cells don't have a shell. And so when I removed the shell, they are now very similar to the cells in our body because they have what's called a cell membrane on the outside. And that's what looks like, they look like water balloons right now. So the outside covering, that cell membrane that looks like a water balloon, is actually still offering protection for the shell and is keeping all that liquid that's inside protected and not from, prevent it from spilling all over the place. So we're going to use these as our cells, and we're going to do some demonstration with different substances to see if we can determine what types of things can go in and out of the cell. So those are going to be our cells. So the way the cell membrane works is it actually is not a solid substance. It's what's known as a semi-permeable membrane. And the best way to represent this is actually to look at something like a strainer or a colander. You can see, for the most part, it's pretty solid, but there are holes spread throughout the surface all around. So the membrane around our cells actually acts very similar to this colander. So if I have a piece of pasta, like this shell pasta, I can't find any holes that are the right shape or size to make this shell pasta pass through it. So if this is the outside of the cell and the inside of the cell, there's no way for me to actually pass this all the way through. But if I have a piece of spaghetti, I can actually pass the spaghetti right through to the other side. And the spaghetti is able to travel into my cell. So the cell membrane works very similar to this. So we're going to see what types of substances we can set up with our eggs and see what is actually able to travel in and out of the cell.
All right, so I'm gonna set up my experiment. I've got 7 cups here to represent the different variables that I'm going to put my eggs in. The one in the middle here is my control. So we're gonna leave that one alone, just put an egg in and leave it alone to see what happens just by itself. And then I've got 6 different things that I'm going to adjust and put 1 egg in each one to see what the impact is. I'm gonna weigh my eggs before I put them in and then after to see if there's any change in their weight or their mass. And that'll tell me if anything was able to go in or out of the cell. So the things I decided to test, because I had them in my kitchen, I've got corn syrup here, which is like a sugar syrup. I've got plain water. I'm gonna make a saltwater solution. I'm gonna make a water solution, but I'm gonna add some red food coloring. I'm also gonna make one that's blue. And I'm going to make a sugar water solution. So similar to corn syrup, they both have sugar, but this one's just gonna be table sugar and water dissolved in, so it's a liquid. This is more thick, so I'm gonna see if there's any difference. And I'm gonna mix blue and red food coloring in this one to make purple. And then because this is Easter themed, I'm gonna put the yellow food coloring in one of them and the green as well, so we get a nice colorful mix. And I want to see if any of that food coloring is able to make it inside our cells. So let's set up our experiment. I've got a scale here, it's measuring in grams, so I can measure the before and after. I'm going to start with my control egg, so I don't have to do anything to that one. And this egg is measuring 94 grams. So I'm just going to put that one in my control cup very carefully, and that one's ready to go. Okay, so now I'm going to make my solutions. I've decided, just for my test, for my experiment, I'm going to put in a tablespoon into my sugar water, which is going to be that one. And I'm going to put a tablespoon of salt into my salt water one. And then I'm going to mix them up really well to hopefully dissolve all of that out. Okay, and then my corn syrup one was already done. And so I need to mix up my sugar water. And I'm going to get another stick and mix up my salt water. And the sugar's dissolving pretty good, almost clear, meaning it's all been dissolved. There we go. Now my salt, having a little bit more of a hard time dissolving in. So we're going to stir it as much as we can and get as much of it dissolved as we can, and that's okay. All right, so there's my salt water, and I'm going to add my food coloring. So I've decided to add 2 drops. See if I like that color. That's nice and blue. I'm going to add 2 drops of red. Give that a stir. And then let's make our plain water yellow. Okay. And we are going to make our salt water green. This is kind of a fun, take on dyeing Easter eggs that are typically hard boiled. And when you do that, you're just dyeing the shell. But we removed the shell, so now we're going to see if we can dye the inside of the egg. And then I would like to make my own color. So I'm going to do 1 drop of red and 1 drop of blue. Any guesses what color that's going to make? It should make purple. All right, so there we go. So now I've got all of my solutions ready to go, and I've labeled them on the outside of the cup so I don't forget. So I take all the stirring sticks out and weigh my eggs. So let's turn this back on. All right, so this egg weighs 100 grams exactly. We're going to put that in salt water. Okay, just fits. So let me write that down. 100. Make sure our scale is at 0. The next egg is 92. I'm going to put that in my plain water. Okay, water 92. My next one is 94. So I'm going to put that in my red water. Next one is 89. I'll put that in blue water. Okay. The next one is 82. It's going to go in my purple sugar water. And the final one for our corn syrup is 79 grams. And I'm gonna put some more corn syrup in there so he's covered up. All right. So I'm gonna set these on the counter. I'm gonna do a time lapse so we can watch them. And I'm gonna leave them for about 24 hours and come back and make some observations in 24 hours. All right, so our eggs have been sitting for a little over 24 hours now. So now we're gonna take them out, weigh each one, and that will tell us whether the mass has changed of our eggs and whether any substances have gone in and out of the cell. So we're gonna start with our control that is not sitting in any solution. And we are gonna weigh it to see if there's any difference. When we weighed it the first time, it was 94 grams. Now it is 87 grams. So that means that something was able to get out of the cell, most likely for evaporation. I didn't have it covered up with anything, and the bottom of it is actually a little bit wet. So it does seem like some liquid just naturally left through the membrane of the egg and probably evaporated out into the air. So that is kind of our standard. So normally just sitting out, it seems like about 7 grams of weight was lost. Okay, so we're gonna use that now to compare each of our variables that we did. So the ones right here are all ones that are sitting in water. We did add different colors just to kind of make it fun and mix it up a little bit. And it's a good thing I did because one of them actually did break at some point in the process. And so the yolk is still there, but you can see the outer part of the egg has definitely cracked and it's just kind of floating near the bottom. So I'm not gonna weigh that one, but I have 2 other ones that I can weigh. The other thing, when I pull these out, we wanna look for, not only are there possibly liquids that are coming in and out, but the reason we put some color, some food coloring in, is to see if any of that food coloring was able to get in or into the cell, into the egg. So I'm gonna start with the red one, and I'm gonna do my best to take it out. I'm gonna dab it off a little bit, and just so I get most of the liquid from the cup not being weighed. And I'm just going to dab a little of this moisture off, and you'll see I have a pinkish red egg. So we know that the food coloring was able to go into the egg, and oops, it's turned off. This egg now weighs, it started at 94 grams. It now weighs 98 grams. So it went up by 4 grams and it turned red. So that's pretty interesting. It seemed to have absorbed some of the water, and it definitely absorbed the food coloring. Not all of it, there's still some pinkish red left in there, but it definitely absorbed some because my egg is now colored. So that's interesting. Let's see if the yellow one has a similar result. So my yellow one started at 92 grams. Let's take this one out, take out some of that liquid. And the yellow egg is now yellow. Just like the inside, we know the yolks are kind of yellowish, but now the entire egg is yellow as well. All right, so let's see what our yellow egg now weighs. See if it's similar to what happened with the red. The yellow now weighs 87. And it started at 92, so it actually lost a little bit of weight. So that's pretty interesting, but also yellow. So we know for sure that food coloring can cross an egg's membrane. Okay, now looking at the 3 we have left, I'm not going to weigh the blue one because that one broke. Looking at the 3 we have left, we've got salt water here in green, and then these 2 are both sugar water. So the corn syrup is very sugary. There's a very high concentration of sugar, and you can probably tell from just looking at it, it's kind of starting to shrivel up and definitely looks smaller. But I'm gonna weigh it and see how much smaller it got. This one has table sugar mixed in, dissolved in with water. And I can't tell right off the bat if that's any different in size, but we will find out. So let's start with our salt water. Let's see if this one's green. Yep, sure enough, it's green too. So all of our colors have been able to travel across the membrane of the egg. I'm gonna dry it off. Now this one started at exactly 100 grams. And now it's at 104. So it's a little bit bigger. Not too much, but it definitely got bigger. Now, one thing that you could play around with if you wanted to just investigate one variable, like the salt concentration, is have a series of cups that have different amounts of salt in them. I didn't put a terribly high amount of salt in. It was about a tablespoon. So the one thing you could do to see if that might have had an influence on it is do different amounts of salt in each cup and just test the amount. So that would be something I might do in the future to see if it actually has, it seems like it has an effect, but maybe I needed to add a little more salt. So that might be the next experiment I might try. All right, let's do our sugar water in purple here. Yeah, there's a purple. So the interesting thing with the purple, I made purple by combining red and blue food coloring. And so I was kind of curious to see if I was gonna get that mixing of the purple, or if maybe there would be little spots of red and spots of blue, like maybe they separate out. But nope, it definitely looks very purple. So let's see what our sugar water purple egg weighs now. It started at 92 grams. It's now 86 grams. So it went down a little bit, not too much. Again, I could also do another experiment. You notice that this water is almost completely clear. So almost all of that food coloring is now in the egg. My other colors, there is definitely still some food coloring left, but this one, it's almost made its way all into the egg, which is pretty cool. So again, my next experiment could be different amounts of sugar to see if there's a point where it makes a really big difference on the size of the egg. All right, our last one here, the cornstarch. This guy looks really small now. It's almost like it's got just, you know, a lot of extra membrane there and not enough filling. So I'm gonna definitely predict that the weight of this one has probably gone down. It did start at 79. Let's see if I can get it out in one piece, would be good. Drain out some of this cornstarch. That's our corn syrup that's left in my little thing here. Let's dry it off a little bit. Don't break it. All right, so, and look at that. It's hardly got any liquid left in, probably just that middle orange yolk part. Okay, so I'm gonna set this in very carefully. There we go. It now weighs 38 grams. So it cut its weight in half in that corn syrup. So because corn syrup is basically very high dose of sugar, That's why it might be really interesting to play around with sugar and vary up how much table sugar you put in, because our one with the table sugar with just a tablespoon didn't really have much of a change, but in this more concentrated sugar syrup had a huge change. All right, so now let's go draw some conclusions and figure out how we might take this one step further in your investigation at home. Okay, so here's our data written out now on the screen. And I went ahead and did a little bit of calculations to figure out the difference. So when I subtracted our before weight from the after weight, that's our difference. So a little bit of math involved here. And what I notice, so you kind of look at what you see. What does the data tell us? We had observations with our eyes, looking at things like the size, the color. We also have our number data here. So we want to use both sets of data, our number data and then our qualitative data of what we saw. And so what I can see, what I would notice is that no matter what, the eggs will kind of lose a little bit of water, a little bit of volume, okay, some weight, just sitting out in the air. And that's probably due to some evaporation happening. Just like if you leave a bucket outside, eventually that bucket loses some water to the air. And then the biggest number, the biggest difference was definitely our egg sitting in that corn syrup. So remember, corn syrup is very heavy on sugar, okay? It's very sugary. And so that really high concentration of sugar lost 41 grams, and that's more than half of its weight just in that 24-hour period. So I want you to think about, what do you think it actually lost? So when you have weight, when you have a mass, what did the egg lose? How did it lose that weight? What was that weight in the form of? So I want to see if you have any ideas. And then I move into my sugar water, which didn't have as much sugar as the corn syrup, but I put a little bit, about a tablespoon or so of sugar in. It also lost some weight, but it wasn't really that much different than my control egg. Then I moved into my salt water, and what I noticed with the salt water is it actually gained weight. So I want you to think about, okay, how does an egg gain weight? We had an egg that lost weight, and we had an egg that gained weight. So what do you think is actually gaining or losing? How is the egg changing its mass? And then I went into my substances that were just in water, and I colored them so I could see. And they were a little bit mixed, but in general, almost stayed the same. So that's kind of interesting. They almost stayed the same. So that's kind of stable. Remember that stability and change from earlier, those words? So again, think about, what do you think is actually coming into or out of the cell, our eggs, to make them gain or lose weight? All right, so let's take a look now at what's happening. So osmosis is a process that happens not just in these chicken eggs, but also in the eggs, in the cells of your body, and any living thing that's made of cells. Remember that that strainer, that colander I had that had the holes in it, and it seemed like, okay, some things like the spaghetti could pass right through, and other things like the bigger pasta noodle couldn't. It was too big. So what's happening with our cell membranes, they're semi-permeable. So semi meaning a little bit, like half. Permeable means things can go in or out. So if you thought about what might be going in and out of our eggs, if you said water, you're correct. So water is actually able to go in and out through the cell membrane, whether it's these eggs or the cells in your body, and it can go freely one way or the other way. Now, it's not random, but what it's trying to do is it's trying to reach an equilibrium. So equal meaning it's right in the middle. And so what it's trying to do is trying to balance out the concentration of the things around it, like the sugar and the salt and different things that are inside and outside of your cells, nutrients. And so what the water is trying to do is balance everything out. So what's inside the cell is the same concentration as what's outside the cell. It's always trying to reach that balance. And water is able to freely move across the membrane either way. So with our cells that were in, our eggs that were in the corn syrup, where it lost all of that weight, which way do you think the water was moving? Was it moving out of the cell or into the cell? And if you said it's moving out of the cell, you're correct. When water leaves the cell, the cell will shrivel down a little bit and it's losing mass because water has a mass. And so the water is trying to balance out the concentration of that really heavy sugar concentration. And when we put it into the cup with all of that corn syrup, that really sugary syrup, now there was so much sugar outside the cell, the water is trying to balance that out because it does have some sugar inside the cell, but not very much, not as much as as what was outside the cell. And so we were able to actually physically see that there was a lot of water lost to the outside of the egg because it's trying to balance those concentrations out, the sugar outside of the cell, the sugar inside of the cell. Now, the opposite was kind of happening with our salt water. And so what would be really interesting if you set this up yourself is to do different amounts of salt in water and different amounts of sugar. And you might be able to see those two different things happening at the same, with your different setup that you have. So osmosis is about water moving across that semi-permeable membrane because the sugar and the salts are not able to just go right back and forth any way that they want. They actually have to go through those holes like what was in that strainer. They have certain pores that they're able to go through, little channels. But they have to kind of be helped out. They can't just pop in and out whenever they want. Water can. So water is always trying to balance everything out when the other molecules are not able to, like the sugar and the salt. Now, what was really fun is that we put food coloring in to our water when we first started, and then we put the egg in. And the egg started out with no color, just the white. And so when we put it into the food coloring water and then let it sit for 24 hours and take it out, that food coloring had actually gone across the membrane into the cell. So that's really cool. You can see that there's definitely things that travel across just by the fact that our eggs were able to change color by sitting in a liquid of food coloring. All right, so your challenge now, like I said, this is not the end of the experiment. We've got to take it one step further and figure out what happens when we put our eggs in different concentrations of salt water or sugar water or whatever you'd like to try out, and see if you can explain how either these osmosis dyed Easter eggs or maybe ones that you set up at home, how water, remember it was the water that was traveling back and forth, how it travels across membranes, just like in our own cells, and see if you can explain it to somebody at home and show them the wonderful knowledge that you now have about how osmosis works in our cells.